Alright everyone, while we're waiting on our brine shrimp to hatch, let's look at our microscope. What you have here is called a compound microscope. The word compound means multiples. It means that it's because there are two different lenses that you're using for this one. The one at the top is called your ocular lens. You're going to take the cap off, and just to let you know, your microscope may not be exactly like this microscope, but it's going to be similar. It's going to have similar parts. So anything that we do with this microscope, you should be able to translate into your own. Now, take off the cap at the top. You have two different ocular lenses. These are the ones that are the eyepiece. If this is not clean for some reason, what you're going to use, do not use paper towels or napkins or anything that's a paper product on it. What you're going to want to use is a piece of clean cotton that you can wipe your lenses like that. If for some reason the lens does not come clean by just wiping it with the cotton, then what you're going to do is you're going to put a little bit of alcohol onto the cloth, not onto the lens. Onto the cloth, wet it with a little bit of alcohol, and then wipe it and let the alcohol dry off of it, and then put it in your microscope. That will make it nice and clean then. So this is your ocular lens. We put the 10x ocular lens. You also have a 15x ocular lens. These are going to be this one. The 15x is obviously slightly more powerful than the 10. Now moving down from your ocular lens, we're going to have the focusing knob. So this is your focusing knob. Turning this adjusts the lenses so that you can see, you can adjust the focus. You're going to need to move this up or down to find your focus for whatever you're looking at. Down here, you have your revolving nose piece. Now your revolving nose piece has on it three different objective lenses. You have a scanning objective lens, you have a low power objective lens, and you have a high power objective lens. The scanning one is the shortest one. It's a 4 times power on that one. So it's a 4 times power on that one. It's your shortest lens. That's the one you're going to use when you put a slide on here. Because when you put a slide on, what you're going to do is you're going to slide your stage clips. This is your stage, down at the bottom. Slide your stage clips to the side. Set your slide on here, with the right here in the hole in the middle of the stage is where you're going to want your main thing to focus. Take your clip, slide your clip back up on here. Do not pick it up and snap it. That will break your slide. Take it from the side, move it across it very gently, and you'll have your slide on there. Now, you focus then, put your eye to the ocular lens, focus with the focusing knob, and move your slide around. You may need to move your slide around a little bit to find what you're looking for. But once you find it with your scanning objective lens, then focus in, get it in really good focus, then you're going to move to your second lens. Your second lens is your low power lens. Your low power lens is a 10x, so it's a 10 times focus. It's a 10 times magnification. Look through your ocular lens again, focus a little bit more because you're not going to have it quite in focus, but you should be close. So focus it in, get it really sharp focus, that way you can see what you're looking at. However, if you want to get even closer to it, then you go to your high power objective lens. Your high power objective lens is 40 times the magnification. So, for the total magnification, you multiply your ocular lens times your objective lens. So a 10x here and a 40x here means this one is running 400 magnification. In the case of your low power lens, it's a 10x lens and a 10x lens, so that would be 100 magnification. So now you're going to focus it again, but be very, very careful. The high power objective lens is very, very close to your slide. If you focus too much, you'll push the objective lens into your slide and crack it. So be very, very careful whenever you're focusing with your high power lens. You're only going to want to move just slightly. If your specimen was in focus with the low power lens, you should be very close to focused with the high power. You may need to move it just a tad, but not much. And there is a safety feature on most of your microscopes. There's a little screw here. This screw should be all the way in because it's to keep this lens, it's so you can only turn it down so far to help keep this lens from cracking your slide. But do not count on it. Be very, very careful when you're focusing with the high power lens. Start out focusing with a scanning lens, then move to the low power lens, and then finally to the high power lens, so that when you put your high power lens there, it should almost be in focus. Now, we've covered the stage clips, we've covered the stage, we've covered focusing, we've covered your nose piece with your objective lenses, and your ocular lens. Oh, the other thing is, if you want to get a little bit more focus, you can change your ocular lens from the 10 to the 15, which will give you half again in focus. So the 40, the high power with the 40x, which was 400 with the 10x lens, is going to be 600 with the 15x lens. So that gives you a little bit more magnification if you need it. So you can switch out the two lenses there. Now the last thing that we need is light. Because this is a light microscope, it only works by light coming through whatever specimen you're looking at. You can't look at a rock or something with this. You have to look at something that light is coming through. So that's why it works great with liquid things, so drops of water, blood, anything like that that's a liquid thing that you can see light through. But, okay, or what we're going to do, to get light through, we're going to have to use this little mirror here. What you do is you have a light source, a lamp or the sunlight or something that you can reflect the light up through the hole in the stage through your specimen so you can see it. Now, this works great, but you may not want to use the mirror. Most of your microscopes will come with a light source for them. Now this light source should have your switch over here that turns it on and your light source. Inside it, if you check, is a battery case. So if it doesn't work, if it's working, don't mess with it. But if it doesn't work, take out your battery case, replace the batteries, and it should work fine then. But it should work fine to begin with. And if it works fine to begin with, don't mess with the batteries. But if your batteries go out, you need to replace them. So you've got your power switch here. You've got your light here. All you do is flip the switch on and ow! 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 Excuse me. Make sure you're pointing the light in the right direction. So you've got your light source here. You can turn it on and off. To put this in, what you're going to do is you're going to grasp your mirror, pull your mirror out of the slot, replace it with your light source, make sure it's pointing up through the hole, and you're ready to go. So this means you're all ready to go to look at your slides as soon as your brine strip are finished. We'll show you how to prepare a wet mount for that so you'll be able to see them. So that's it. Make sure that when you are storing it, you store it with the scanning lens in place. Make sure that when you carry it with two hands to support it. The bottom hand supports the weight of it. The other hand just keeps you from dropping it. And anytime you're not using your microscope, keep it covered. You should have a microscope cover, some kind of a plastic cover that you can put over it. That should have came with your microscope. However, if it didn't, just have a towel, some other thing that you can put over the top of it while you're storing it without the light on. While you're storing it, that just keeps dust out of your lenses. So there you go. There's your microscope. You're going to have fun with this.